Okay, so today we're going to start our discussion of genome-wide selection within the context of the Regression BLUP package, the same package that we used yesterday for genome-wide association studies. And so I'm going to start by talking a little bit about the difference between association analysis and genomic selection, which involves estimating a breeding value. So in genomic selection, we're trying to predict a breeding value for individuals based on their genotype and kinship. So a breeding value is the part of an individual's phenotypic value that is due to additive genetic effects. It's the value of an individual as a parent. The approach that we're going to take is we're going to assign a breeding value to each marker in our data set, regardless of whether that marker is significantly associated with a trait or not. Then we can create a genomic estimated breeding value, which is essentially the sum of all those individual marker effects. Okay? So in order to give you an example of what we're doing, I have this naive thought experiment that I'm going to walk you through. Okay? So what I'm going to do here is I'm going to compare stepwise with multiple regression. Okay? And so here, Fit1 is a model that you could all write. Right? We're using marker as a random effect to look at its value, or it's an effect with respect to a specific phenotypic trait. Okay? So if we have a situation where we have 5 markers, we can walk through a series of 5 individual random effects models in order to estimate the effect of each marker. Right? Or alternatively, we could write a single model that incorporates each of those markers. Okay? So let's look at this with respect to a specific data set. If we look at the stepwise regression and the effect estimated for those markers, we can summarize it here. Okay? So this happens to be a backcross population, the data that I use, so you only have, you know, what amounts to heterozygote and homozygote. Okay, and you can see what the effect of an allele substitution is for each locus. And the key is, for these markers, we have no effect, right? Whereas if we run everything together as a single model, note that this effect here is slightly lower than this. So in other words, we could say that this might be an overestimate. We have some effect here, so this is an underestimate. Right? And the same is occurring here. Right? So in other words, when we use a single marker, we're likely to underestimate some markers and overestimate the effect of others. Whereas if we run them all together, we're going to distribute those effects across the genome. Okay? And so this is in a sense a contrast between what we're doing with marker-assisted selection and what we're trying to do with genomic selection. So with marker-assisted selection, we're focusing on only a couple of markers that we know are linked to traits that we're interested in. Okay? we're likely going to overestimate the effects of those while underestimating the effect of the rest of the genome. Okay? So in genomic selection, we're going to say we don't care anything about association or linkage. We want to get estimates that are spread across the genome. Okay? So does that make sense to everybody? Right? Okay. So stepwise regression is a single marker at a time, whereas multiple linear regression, we're putting all of our markers into the model at the same time. Okay? The problem is, or a problem arises when we get to the point where we have more markers than observations. So if I have a large population, say 384 individuals, but I have 10,000 markers, I have more markers than observations. And so the model becomes over-parameterized, right? And so the only way to handle that is to use some sort of a correction or a penalty to restrain that model, right? To make sure that the model doesn't explain more than 100% of the variance, right? And so there are different approaches, and this is where the literature has thrived since Musin et al.'s paper, right? There are multiple approaches to constraining the model or putting a correction or a penalty on it. Okay? So ridge regression, lasso, Bayesian approaches, Bayesian lasso approaches, Bayesian ridge regression approaches, right? Those are just a few of them. Okay? What you have to understand is that ridge regression is imposing a uniform penalty on every marker, right? It's like you say, okay, look, we have the possibility of unit variance, so therefore we're going to divide that unit variance by the total number of markers and impose that penalty on everything, right? So it's kind of a naive approach to constraining the model because it assumes that all markers will explain equal variance. And yet we know that's not the case, right? Markers that are in fact linked to traits will explain more variance. Okay, and so these other approaches, like the lasso approach, is used to impose differential constraints on different markers. Okay? All right. So again, this is kind of what we're saying here. So we want to estimate the parameters or the breeding value for each marker in our model. Okay? And so we can use least squares regression approaches with markers considered random. Okay? And I'm unfortunately, I kind of chose the wrong notation here because we're used to thinking of x as the fixed effects, but I'm just simply modeling random effects here. So the way this works is that we set the residual vector is set here, okay, and we solve by minimizing that. In other words, we can use a maximum likelihood estimate of the parameter values in the vector beta to do that. Okay, and the vector beta is given here. All right. So if you go to this URL is explaining the mixed solve function in rrBLUP, and the notation used is given here. Okay, and so the model that mixed solve can create can have both fixed and random effects. Most of the time when people are running ridge regression BLUP for genomic selection, we're not putting in any markers as fixed effects. We're only putting them in as random effects. Okay? But I will give you an example today of how you can put some markers in as a fixed effect if you choose. Okay? And so we're either solving for the fixed effects as a best linear unbiased estimator or the random effects as a BLUP. So the statistical considerations for solving these equations: there must be a solution, the solution must be unique, and the solution must depend continuously on the data. Those are pretty much givens. Okay. So because the predictor variable may be correlated, i.e. linkage, because some markers may explain no variance, there might need to be some corrections. Okay? So these corrections, or statisticians sometimes refer to this as regularization, involve introducing a penalty that puts bounds on that vector. So, you know, if we're talking U, right, which we're trying to estimate as a BLUP, right, that should not be greater than the variance itself, right? It should be restrained. Okay? And so in ridge regression, we're adding a constant to the diagonal of the matrix of coefficients that makes the solution unique, and it shrinks the estimates of marker effects. Okay? So in order to do this, we first have to sample the data so that we can get these estimates and then apply the proper shrinkage. Okay? So this is the key, is that in ridge regression, those effects are penalized to the same extent. Okay? And so the lasso approach, least absolute value selection shrinkage operator, instead of penalizing them equally, will penalize some more than others. Okay, and again, that takes sampling of the data in order to do. All right, so this diagram is adapted from the Hefner, Sorrells, and Janik 2009 crop science paper, and it describes what we're trying to do with genomic selection. Okay, so we start with what's called our training population, and that's a population that we have both phenotype and genotype for. Okay, we use these data to create a genomic selection model which we optimize by cross-validation. Okay, once we have this model, we're going to apply it to material that we only have genotypes for. What does a genomic selection model look like? So when we talk about our genomic selection model, which has been optimized by cross-validation, what I'm really saying is that I have a vector that has, for marker 1, a small positive or negative value. For marker 2, I have a value. For marker 3, I have a value, right? And that this vector is my model. Okay, so this is going to go on for, you know, however many markers I have, right? This is my model. Okay, it's a vector of values associated with each marker, right? And what I'm going to do is, when I get material that is only genotyped with the same markers, right, I'm going to have a marker matrix that I'm going to multiply by a vector of marker effects, and for every individual in that marker matrix, I'm now going to have a single value that is an estimate of the performance. Okay? So my quote-unquote model is simply a vector of marker effects. I'll clean up my workspace just a little bit here. Sorry. All right. So ideally, the way this is working now is I have genotype data here. I multiply those genotypes by my vector of values, and I come up with this estimate of a value associated with those individuals. I base my selections on that, and now these selections will end up in the field where they will be phenotyped. Okay, because I have marker data on them and now I have phenotypic data, I can add this information back here and rerun my model. So over time, I have more genotypes and more phenotypes in this model, so in theory my model is improved, right? Also, as I'm selecting, I'm probably eliminating the polymorphism of some markers, so things are changing here, but this is the retraining process. As I add phenotypes and genotypes, I can add it back in and rerun this. This process of selecting based on genotype is never more efficient than selecting based on phenotype, or almost never more, right? So the only way that the breeding process can be more efficient in terms of gain under selection is if we somehow tie this selection into an accelerated generation time here, so that we get more selections per unit time than we would otherwise. Okay? So genomic selection only makes us more efficient if we somehow tie it into accelerated generation time. All right, so let's move on. Okay, the data that we're going to talk with is going to be trait values, possibly a kinship or pedigree matrix, a marker matrix, or SNP scoring is going to be numerical, proportional and numerical, and implied in this, and this is again another key, we're talking about additive models, right? Well, the additivity is implied by the scoring of the marker, right? So if we score to, so this is you know, one allele, 2 doses, right? It implies that proportional scoring implies that the heterozygote is the mid-parent. Okay? And so when people talk about modeling dominance in their genomic selection, what they really are talking about is changing how you score the markers.